Alright, so hopefully you watched the end of the respiratory system already before moving on, because there were a bunch of little announcements, but I'll make sure those announcements go up on Blackboard. I'm going to continue talking about the digestive system. I have these funny diagrams up here just to kind of talk about the perpetuation of interesting misconceptions people might have about what the digestive system does and what various features do. And so just keep in mind when you're talking about things to people, they might have weird things to say about their digestive system. You know, the more you know. And so again, along with the respiratory system, the digestive system you've covered in lab already, and so a lot of this should be reviewed because we're just going over general functions of things. And so what are some of the general functions of the digestive system? Does a lot of things for us that we are very thankful for, right? And so we ingest food. We move food through our digestive tract. We mechanically process, meaning that we chew it up, we mix it up, various things like that. We chemically digest it, absorb nutrients, and then eliminate anything that we cannot absorb. And so again, I always harp back on why do we have these systems. And so we talked about the respiratory system at the beginning, and I said, why do we have the respiratory system? And usually the most common response is, well, we need to breathe. Okay, well, why do we need to breathe? Oh, right, we need to breathe because our cells are metabolizing. In order for our cells to undergo cellular respiration, they need oxygen. In order for our muscles to work, we need oxygen, right? We need to produce ATP. So we need oxygen, we need glucose, right? We need sugar products, and so that's why we eat, is because again everything boils down to our cells are alive and they need to undergo cellular respiration. We need to create ATP and all of that fabulousness. We need to consume food in order to get the glucose, but we don't typically just eat plain glucose, right? It's not really all that good for us and could lead to diabetes. So all the systems are interacting, talking about the endocrine system, but we need to think about the fact that when we consume food, most food items that we consume are not in the proper form for us to directly use. And so what we need to do is actually be able to break the food down and get it down to its basic components. Those basic small components are what we are able to move across our membranes and our intestines, right, and absorb into our bloodstream and move things around. And so molecules need to be small enough that we can absorb them directly across our membrane, but they also need to be in a form that's usable, right? So if we consume a plant, those amino acids in that plant may or may not be in the usable form that we want or can use. And so the digestive system is really important in breaking that food down into those basic components and allowing us to kind of use them as those building blocks after we move those building blocks across our membranes into our bloodstream. And so I always think about kind of how does your digestive system work, thinking about, okay, well, if I were to consume my mom's macaroni and cheese, which is super delicious, right? What are the basic things that I'm going to be absorbing from that? Well, there's fats from the cheese and the butter and all of that, things that make it delicious. There's also starches, so we're going to be ultimately absorbing monosaccharides and disaccharides and whatnot. So simple sugar molecules from those starches eventually after everything gets digested. And so we have fats, sugars. We also have proteins, right? Proteins from the milk and the cheese and things like that. And so thinking about what the digestive system does, the best way to start is just thinking about, okay, what would happen if I were to trace a molecule of food through the digestive system? What's happening where and how? You can even use this trick for the respiratory system. So thinking about how the respiratory system works. Follow a molecule of oxygen from the external environment all the way down into the lungs, into the capillaries. What are the structures that it needs to pass by as it's getting to the lungs, right? And so it's just a really nice way to create paths in your mind to help you learn the various different features and structures in each system. And so I'll stop digressing talking about the respiratory system and get back into the digestive system. And so let's talk about the major components of the digestive system. And so this is just a broad overview, and then what we're going to do is kind of talk about the highlighted parts. And so of course, digestion begins with ingestion of food. And so we ingest food hopefully through our mouths and not through our nose unless we're laughing hysterically at something in our lunch table, maybe when we're in, like, I don't know, 5th grade. But we use our tongue and teeth to start chewing up the food, and then we use the oral cavity to kind of move the food down to the esophagus. And so we also have salivary glands. These are the salivary glands that you had to know in lab. So the parotid gland is the big one on the side of the cheek. And then sublingual, as it sounds, is under the tongue. Submandibular is just under the mandible. These provide salivary amylase to start the starch digestion process. Food moves past the pharynx. So remember, the pharynx is that shared area, so the oropharynx, laryngopharynx, and then into the esophagus only, right? The epiglottis doing us a favor by kind of keeping that food going to the esophagus. And so food moves into the esophagus, past the stomach for mechanical, chemical digestion, storage, and a tiny bit of absorption and things like that. And then we have digestive aids from the liver, the gallbladder, and the pancreas that are producing digestive enzymes that get dumped into the small intestine. That very first part of the small intestine is really important in kind of finishing the digestive process. And so the small intestine is broken down into the duodenum, which is the very first region right off of the stomach, the jejunum, which is kind of the middle region, and then the ileum, which is the last aspect of the small intestine prior to food moving into the large intestine. And so food moves then, after you have a lot of absorption happening in the ileum and the jejunum, moves into the large intestine. The large intestine has a lot of different features. Major role in the large intestine is compaction, water reabsorption, and things like that. And so we have the appendix, which sticks off of the cecum, and so the ileocecal valve is what joins the small intestine to the large intestine, and so ileo for ileum, ceco for cecum in the large intestine, and so the cecum is that very first pouch that you see here. What's really cool about the animal kingdom is the size of the cecum varies quite a bit, and so if you look at the cecum of a horse, it's really big compared to the cecum of a human, which is actually quite small. Like I said, this part right here is just a cecum, and a lot of it has to do with what we consume, and so we don't have a heavily we do have a relatively plant-based diet, and we cannot survive without plants on this planet, which is why you should all care about plants and love them. But we don't consume them like horses or cows and things like that, and so a lot of extra digestion is happening in the cecum. And so anyway, I digress, and back to teaching. So here's the cecum: ascending colon, transverse colon going across, descending colon, and then the sigmoid colon here, kind of the S-shaped, leading into the rectum. And then last but not least, you have defecation through the anal canal and anus. So you should know and love all of these features, and like I said, we're going to go through each one of these a little bit more detail individually. And so your book does a nice job of kind of providing a broad stroked overview of kind of what we're going to be talking about. And so if we talk about your mom's favorite macaroni and cheese, right, we ingest that, the food initially gets chewed up and masticated, and we have initial starch digestion with salivary amylase. It moves down into the esophagus and then into the stomach, where we have the start of protein digestion, and so that's where you see those droplets of chemical digestion. And then continuing with chemical digestion in the small intestine to finish off fat digestion, starch digestion, and protein digestion. And then primarily absorption of those macromolecules, so absorption of sugars and amino acids, nucleotides, and small fat molecules get absorbed into the blood vessels or lymphatic vessels in the ileum and the jejunum. And then any waste material that's not absorbed and not fully digested enters into the large intestine for kind of compaction, absorption of water, and again, some vitamins are reabsorbed. Again, broad overview from the previous slide and this slide. We're going to go into it in more detail just like we did in the respiratory system. But before we can do that, we need to talk about the histology of the digestive tract just for a second. And so talking about the four major layers of the digestive tract, and so you can find all of these layers throughout your digestive tract, which is why we talk about them, and they're really, really important. And so you can see the lumen here, which is the opening. We talked about lumens when we were talking about the cardiovascular system. It is the open part of a tube when you look in it. And so here would be the lupin, sorry, not lupin, the lumen. And then the mucosa is this very first epithelial tissue or endothelial tissue surrounding the lumen. And so the mucosa lines the tract of lines the inside of the digestive tract. It is a mucous membrane, and so it does produce lots of mucus. Your digestive tract has mucus throughout it, and we are so thankful for that, because otherwise our food would scrape along our epithelium the entire way down. And so without the mucus, it would cause a lot, consuming food would cause a lot of damage. The submucosa is this layer here, and so what you see is it has lots of blood vessels, lymphatic vessels, and nervous tissue, and so it's dense connective tissue, lots of different types of vessels, blood vessels, lymphatics, nerves, and then there are some exocrine glands, and so remember, exocrine glands are these ones that are going to be contributing to the digestive process, and as opposed to the endocrine glands. We have then the muscularis externa. The muscularis externa is made up of circular and longitudinal muscles. That's why you can see on the diagram there's kind of two different layers of the muscle tissue of the muscularis externa. And so these are layers of smooth muscle, which means, like again we talked about before, you cannot will your body to digest your Thanksgiving dinner faster than it will on its own, even though we would love to because we're really full. And so it is smooth muscle, which means it's controlled by the autonomic nervous system. This muscle tissue, again, all of these layers are present throughout your digestive system. And so the muscle tissue is really, really important for that peristaltic movement of moving food throughout your digestive system. And then last but not least is the outside, or serosa or adventitia, or adventitia, sorry. It's the outermost layer, kind of isolating it from everywhere else in the body. And so just to remind us of the functions, because I can't fit everything on one slide. And so the mucosa is that innermost lining most adjacent to the lumen. It secretes mucus, digestive enzymes, and hormones. It is involved in absorption depending on the structure that it's in. And so it's not really involved in absorption if we're talking about the esophagus, but it is if we're talking about the ileum and things like that. It provides protection against disease because it is that first line of defense of preventing pathogens to get across. The submucosa provides oxygen, removal of carbon dioxide through the cardiovascular tissue. We have lymphatic vessels, so collecting that lymph tissue, as well as lymphoid follicles. So I talked about the Peyer's patches that are surrounding the intestinal region, so a lot of immune surveillance there because there's high potential for pathogens to move across. There's also neural supply to the area. Again, the muscularis externa is responsible for the churning action and peristalsis, so the churning action that you see in the duodenum and then also in the stomach, primarily in the stomach, and then also peristalsis that you see throughout the entire digestive tract. And then it is also responsible for those fabulous important sphincters. So we'll talk about the pyloric sphincter, which is involved in regulating movement of food from the stomach to the small intestine. We'll talk about the anal sphincter that we all really, really appreciate, and there's sphincters throughout our body that we really need to, appreciate. And then last but not least, the serosa or adventitia makes up the visceral peritoneum. Okay, done with histology. So let's go back to kind of digestion. And like I said, we talked about the overview, and so here we're going to kind of talk about a little bit more of the nitty-gritty detail. And so here I will also have check-ins as we move past kind of major landmarks. So there'll be a check-in right after we finish everything with the stomach. There'll be another check-in after the small intestine. And last but not least, the large intestine is when you know we're almost done. And so in terms of where does digestion start, and so if we're talking about, eating my mom's macaroni and cheese, the irony of this example is that I'd actually am kind of lactose free, so I no longer eat my mom's macaroni and cheese, but I still have a fond memory of it. And so just go with me on that, and I apologize to those who are lactose intolerant, and if this elicits bad memories. I'm sorry, but I digress. All right, so where does digestion start? We started off talking about in the overview. It starts in the oral cavity. And look at all these amazing features of the oral cavity that you know most of, right? Very familiar features like the hard palate, the soft palate, the uvula, that fabulous thing that shakes back and forth when you go, ah, and you look in the mirror. Again, weird things that people do when they're looking in the mirror. The tongue, look how cool the tongue looks, right? Look at that muscular tongue. It's so interesting. Obviously, tongue very important in communication as well as chewing and swallowing and things like that, and manipulating food to move it in the right area. Here's that fabulous epiglottis flap that we were talking about. So oropharynx, laryngopharynx, the esophagus down here, trachea down here. Last but not least, we have things like the lingual frenulum. Lingual frenulum is significant when you talk about babies having tongue tie. It means their lingual frenulum is a little bit more taut or short, and so they can't move their tongue as much, and so it actually affects how they feed and things like that. This is just kind of, an alternate view of things. And so oral cavity, so what's happening in the oral cavity? So think about what's happening in the oral cavity. And here's a fabulous picture showing how the epiglottis bends over the trachea as the food moves down through into the esophagus, into the esophagus. And so what is happening in the oral cavity? We have sensation, taste, right? So gustation, one of the fabulous special senses. We have mechanical digestion through teeth and tongue, mashing the food and masticating it to break it down into smaller bits, which is really important because it's really hard to swallow an entire cow down our throats, right? So we need to chew up bits and pieces. Also, that mastication increases the surface area of the food, which makes the enzymes work much more effectively. And so we do have chemical digestion. So salivary amylase, we talked about those salivary glands, the parotid, the sublingual, and submandibular. These all release salivary amylase, which begins the start digestion process in the oral cavity. And so we do start chemical and mechanical digestion happening in the oral cavity. And so that initial churning and mixing of the food and chewing it up and mixing it with the salivary amylase, that food then becomes what's known as a bolus. That's what you're seeing in the diagram. It's just a chewed up, mashed up ball of food. And so that moves down into the esophagus. And so again, another major role of the oral cavity in that tongue is helps move the food into the posterior aspects of the oral cavity to move the food down into the esophagus. And so major important process. And so just a reminder of the salivary glands. So I already just talked about those different ones. They secrete amylase, which if you see anything with the word ACE at the end of it, that should make you think, oh, it probably is an enzyme that breaks something down. And so amylase starts the starch digestive process. And so that's all you're seeing in this diagram. It's kind of highlighting the parotid, mandibular, and sublingual. And so how does food move? How does food get from the mouth down to the stomach? It moves through peristaltic waves. And so what happens is we have contractions of these muscles in those circular longitudinal muscles that squeeze. And as they squeeze here, it causes the food to move in this direction. And then these muscles here will squeeze. So these ones will squeeze, and that causes the food to move further down and further down and further down. And so peristaltic movements, we see these throughout. For those of you that are starting to look at urinary tract stuff in lab, remember that there are peristaltic waves that help move the urine from the kidney down to the bladder. There's also peristaltic waves that help move the sperm through the urogenital tract. There's also peristaltic waves that help move the egg down the fallopian tubes. And so it is not necessarily a unique feature to the digestive tract, though it is cool and fun to say. All right, so how does food get from the oral cavity to the stomach? It travels through that fabulous muscular tube, the esophagus. The esophagus is specialized for food only, moving down. It is not involved in chemical digestion, or it's not directly involved in chemical digestion or mechanical digestion. All it does is move food down. And so no mechanical or chemical digestion, also no absorption. And so if you relate this back to the anatomy or histology of the esophagus, it has a very thick muscular layer and stratified squamous epithelium. And so where else have we seen stratified squamous epithelium is, hopefully you're thinking, your skin, right? And so it's no surprise that we have that there. We want to have kind of a relatively tough tissue that the food is going to pass through, and so that stratified squamous epithelium kind of helps protect it. And so that's what you're looking at this diagram. Again, you can see the lumen of the esophagus, and so here it's kind of collapsed looking because there's no food in it, right? And so as there's a large piece of food moving through it, it kind of expands to accommodate that food. And so here you can see the epithelial tissue, and then the muscular tissue all the way around it. All right, so food moves from the esophagus to the stomach. And so that's what you're looking at here, the esophagus up here. And so I'm going to talk about the anatomy of the stomach first, and then get into a little bit of the physiology and some of the microscopic anatomy of the stomach. And so the esophagus dumps food via peristalsis into the stomach. The very first region of the stomach, just around the esophagus, is the cardia. And so if you think of heartburn, people have heartburn, it's often due to acid reflux, where we have acid kind of going from the stomach back into the esophagus. And so acid reflux and heartburn should make you think of the cardia region, because that's where a lot of the damage happens. So the cardia region, and then there's the fundus, which is kind of the hump, the body. The pyloric region, which is that very last region leading up to the pyloric sphincter. It is sometimes thought that there's a cardiac sphincter up between the esophagus and the stomach. Whether it's a true sphincter or not, I think is still up for contention for whatever reason. But the pyloric sphincter is very, very, very important because it is closed to allow for digestion to happen in the stomach, rather than just letting food flow through continuously into the small intestine. We need time, we need residence time in the stomach to allow for efficient breakdown of the bolus into what's known as chyme, so mixing with digestive juices to start the protein digestive process. And so that pyloric sphincter kind of holds the food in the stomach until it's ready to move on to the small intestine. And so that pyloric sphincter is really important. The duodenum here, so again, the anatomy of the stomach, I include the duodenum. The duodenum is the very first part of the small intestine. So remember, even though it's on the slide, the duodenum is part of the small intestine, not the stomach. But it is that very first part, and it is very important in that connection of movement through the stomach into the small intestine. And so we have the greater curvature and lesser curvature, just kind of anatomical landmarks. And then last but not least, we have the rugae. So the rugae are the folds. So if you open up the heart in the lab, you would have seen folds in the stomach. If you open up the, If you open up the stomach in the lab, you would see the folds in the stomach. You'd also see them if you were to open up the stomach in your cat. The folds are simply because when your stomach is empty, it kind of collapses on itself. When it's full, it opens up. And so the folds are a byproduct of it when the stomach is empty. When the stomach is full, you do not see those folds at all. They do not play any role in increasing surface area at all. We talk a lot about surface area in the digestive system. The rugae are simply to allow for expansion of the stomach. And so I'll talk about it again on the next slide when we talk about kind of functions of the stomach. But it's just a reminder, and I'm going to harp on this again and again, because there are features in the small intestine to increase surface area to allow for better or more efficient digestive process, more absorption, things like that. The rugae in the stomach do not play a role in increasing surface area. It's just to allow for expansion of the stomach. And so what is actually happening in the stomach? There are lots of different things happening in the stomach. And so you kind of break it down into 4 major categories. We have digestion happening, a little amount of absorption. We have storage. So remember that pyloric sphincter kind of keeping stuff in the stomach to allow for some of that digestion to happen. And then we have propulsion. So we have that peristalsis moving food through when after the digestive process has kind of happened. And so in terms of digestion, you'll constantly hear me break down digestion into chemical digestion and mechanical digestion. Mechanical digestion is as exactly as it sounds, like pounding food, breaking it up by pulling it apart and things like that. And so we've all felt our stomach kind of lurch or move around and things like that. And so we know that our stomach is undergoing some mechanical digestion, but there are also a lot of, there's some chemical digestion happening in there that ties into that acid reflux that we were just talking about. And so the mechanical digestion that's happening is those peristaltic waves, so those muscular contractions of the stomach are mixing the food with the gastric juices that are in your stomach. And so what this does is increases the contact of the gastric juices with the bolus of food that has made its way down from the esophagus, and so allowing for efficient kind of contact between undigested material and those gastric juices that can start or kind of keep going the digestive process. And then we have chemical digestion, and so the stomach, as we know, is an acidic environment. So that acidic environment leads to denaturing of proteins. What denaturing is basically it's unfolding them and allowing them to be more easily degraded by various proteases, so proteins that break, or various enzymes that break down complex proteins. And so one of those enzymes is pepsin. And so we're going to talk about pepsin in a little bit more detail, but so it's the combination of the acidic environment and the presence of the pepsin that starts to digest proteins. And so when you're thinking about what kind of chemical digestion is happening in the stomach, you should be thinking about protein digestion. So starch digestion starts in the oral cavity, and it's not happening in the stomach because of the acidity of the stomach. The acidity of the stomach kind of halts the activity of the amylase, and so we don't really have any sugar digestion or starch digestion happening in the stomach, and we don't have any fat digestion. It's just the start of protein digestion. Like I said, there's very little absorption. There is some absorption, so things like aspirins, alcohol, and some drugs. So a few fat soluble substances happen in your stomach, or can be absorbed through your stomach. And so it is commonly a target for drug absorption because if you absorb the drug in your stomach, it gets absorbed faster than if it has to go all the way to your small intestine for absorption. We also have storage until food can be moved into the duodenum. Again, residency time, I'm going to include some residency times for, I don't know, fun for trivia. But residence time can vary depending on what you've consumed, how much you've consumed, and things like that. But again, that pyloric sphincter kind of halts everything. In phys, you'll talk about kind of the hormonal controls of what regulates when and how food is moving through your digestive system. Like I said, last but not least, that peristaltic waves also move the food from the stomach into the small intestine once the pyloric sphincter is opened. And so how is this happening? So like I promised, we're going to talk about a little bit more detail of what's happening in the stomach. And so we have mechanical digestion, so you have muscular contractions, allows for churning of the contents, so efficient mixing of digestive juices with the food that has just moved down. We have chemical digestion, and so the epithelial tissue is simple columnar with goblet cells. I know we get to talk about goblet cells all the time, which is funny because I feel like a lot of you really love them because they're cool. And so the epithelial tissue acts as kind of a secretory sheet. It's a carpet of mucus that protects against acids and enzymes, so there's a lot of different cells in that simple columnar epithelium, not just goblet cells, and so we'll talk about the importance of those cells and their role in chemical digestion. There's also storage, and so those rugae, like I said, expand so you no longer see the folds to accommodate the amount of food that you can fit in. So your stomach can actually accommodate about a gallon's worth of whatever you choose to put in it. This isn't a challenge. Don't try it at home or at school or anywhere. Gross. I've heard of like the milk gallon challenge. I don't think that ever ends well for anyone. It's a terrible idea and makes me sick. And again, size of the stomach is probably going to vary depending on the size of the individual. Again, no empirical evidence coming from my end. But the prominent folds you see are only visible when the stomach is empty. And so this allows for expansion, so not involved in surface area, just expansion. All right, so stomach epithelial tissue. Like I said, I wanted to talk about the simple columnar, and so it isn't just those goblet cells that we're interested in. And so what you're looking at here, you can see kind of the major features that we always talk about for the various different four aspects of the histology of the digestive tract. So we've got the mucosa, the submucosa, muscularis, externa, and serosa. All of those features are to be expected. What's really interesting is the epithelium. We have these what are known as gastric pits, or these deep invaginations with a bunch of different cell types within here. And so these are not actually part of the rugae. The rugae are just folds in general. These gastric pits are always there. They are always there regardless whether the stomach is expanded or contracted and things like that. And so let's look at the cells that are involved in the stomach epithelium. And so the epithelial mucus cells are the very topmost kind of light pinkish that you're seeing here, these guys. They produce a thick layer of alkaline mucus. And so you may be asking, well, the stomach is really acidic. Why would we want to produce alkaline mucus? Well, you don't want your stomach enzymes to digest your stomach cells, right? And so this thick layer of mucus is really important for kind of protecting that epithelial tissue from damage. When people develop, say, ulcers and things like that, we have a breakdown of the layer of mucus, and those digestive enzymes are starting to degrade away the intestinal wall. And so we also have mucus neck cells, which are like that deeper pink that you're seeing here. They produce a thin soluble mucus, also protective. So here's the mucus cells. And then we have two classes of cells that are involved in producing the digestive enzymes. We have parietal cells and chief cells. Parietal cells look like, kind of look like Pac-Man cells. These are the blue ones that you're seeing. The blue ones dispersed throughout here, and so here these are these blue ones. And so what these do is these secrete hydrochloric acid, and so these are the ones that are contributing to the acidic environment. And then the chief cells are the purple ones. These ones are producing pepsinogen, which is an inactive form of pepsin. And so we do have some lipases. Actually, rather you just cross it out and not think about lipases. Lipases are involved in fat digestion, but you're not really getting any much fat digestion happening in the stomach. But pepsinogen is really important because in the presence of hydrochloric acid, it gets converted into pepsin. And so pepsin is the active form of the enzyme, and pepsin is what starts that protein breakdown. And so the hydrochloric acid creates the acidity of the stomach environment, and so it leads to that denaturing of proteins, kind of unfolding the proteins, allowing for that pepsin to come in and kind of efficiently break down some of the proteins and start that protein digestion. And so that hydrochloric acid is kind of, the role is twofold, or the importance is twofold. It's involved in denaturing proteins, but it's also involved in activating that pepsinogen into pepsin. And what's interesting also, when you have pepsin itself can kind of auto-catalyze the reaction of pepsinogen into more pepsin. And then last but not least, you have enteroendocrine cells that are involved in releasing a variety of different hormones, among them gastrin, which helps with regulating satiety. Again, in phys, you'll talk a lot more about when and why chief cells and parietal cells start secreting their various components and things like that. All right. That was a lot. So we've talked about the oral cavity, the esophagus, and the stomach. And so let's pause and take a check-in and think about what's happening in terms of the digestive system up until this point. And so where and how does sugar digestion begin? You should be thinking, okay, well, digestion in general begins in the oral cavity where you have mastication. But sugar digestion really starts or starch digestion really starts in the oral cavity with the release of the amylase by the salivary glands. And so that amylase, so that's the salivary amylase in the oral cavity that starts with the starch digestion. And then the question about, does digestion occur in the esophagus? That should be a kind of a big red light going off in your brain saying, no, 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 no digestion happening in the esophagus. All it does is move food via peristalsis, right? And so there's no absorption, no digestion, no chemical digestion, no mechanical digestion, anything like that. All the esophagus does is move food. And so then all of the following cells are found in the stomach epithelium except. And so we have mucous neck cells. So if you remember, the mucous neck cell is important in mucus production, as kind of seen in the name, which is important in producing a protective layer of mucus over the cells of the stomach. We have chief cells that are producing that pepsinogen that gets converted into pepsin, which is involved in starting that protein digestion. We have simple squamous epithelial cells. Hopefully that kind of brought up a red flag, because remember, the simple squamous epithelial cells are the ones that we found in the alveoli. And so those are not the cells that we're finding in our stomach. We do see parietal, so if we're trying to think of what cells we're thinking of, we're actually thinking of those columnar cells with goblet cells, right? The parietal cells are part of the stomach epithelium. They are the cells that are involved in secreting hydrochloric acid. And so last but not least, what region of the stomach is adjacent to the small intestine? Remember, adjacent means right next to, and so that would be the pyloric region, which runs into the duodenum, which is that very first part of the small intestine. All right, so feel free to pause, take a break, take a deep breath. Just like we normally do in lecture, I would have given you time to talk to your friends about this. And so we'll move on. So where does food go from the stomach? Like I said, it goes into the small intestine. But before we even get to the small intestine, I want to talk about some other structures, accessory structures, like a purse accessory. No, not the same thing. We've talked about accessory structures when we talked about the accessory features of the skin, right? And how funny it is. So when we talk about accessory structures in the digestive tract, so what are those accessory structures? So that should make you think of the liver, the gallbladder, and the pancreas, all structures that you had to identify in lab. And so why are these considered accessory organs? It is because they do not have, food does not directly pass through them at all. And so what happens is these structures all contribute to the digestive process, but food is not necessarily passing through them. And so what you're looking in this diagram, you can see this big blob is, this is the liver, and then the gallbladder, pancreas here, and then this is the duodenum. And so you can see all of these have ducts that dump into the duodenum, and so they're contributing digestive enzymes to the duodenum. And so let's talk about each of these structures a little bit more before we can continue on with what's happening in the small intestine. And so if you look at the major features, I'm just going to kind of animate all of these because for some reason some of them are not animated correctly. And so we have the hepatic ducts, which are bringing bile down from the liver. So if you remember the liver, so one of the major functions of the liver is to produce bile, which we'll talk about. And so the hepatic ducts here. And then the gallbladder will store that bile. And then upon release, the bile will travel down the cystic duct, down the common bile duct, and then be released into the duodenum. The pancreas here, we have the pancreatic duct ultimately dumping pancreatic juices and whatnot into the duodenum. And so let's talk about each one individually. And so what is the function of the liver? Many of us have talked about the liver. We've talked about it before. We talked about it in the context of the lymphatics, that if we were to have to remove the spleen, the liver would start to take over for some of the functions of the lymphatic system. And so the liver is made up of 4 lobes, left, right, caudate, and quadrate. And the major functions, it is a fabulously very important structure, which is why you get a transplant and you can't live without it completely. And so it does filter and process nutrient-rich fluid. And by nutrient-rich fluid, I mean blood. And so anything that is absorbed across the epithelium in the small intestine, either going directly, that goes directly into the blood supply, first goes to the liver for purification before it gets circulated throughout the body. It also stores glucose as glycogen, as we know, because the liver acts as a target for glucagon, right? And so when our blood sugar is low, the pancreas releases glucagon, targets the liver to release or convert glycogen into glucose to help bring up that blood glucose back up to normal. It also is involved in storing vitamins. It is very important in detoxification. Again, this ties back into what I was talking about with the filtering of blood. The reason why you often see cirrhosis of the liver associated with alcoholism is because there are oftentimes many impurities in alcohol, and so those impurities get lodged into the liver, causing cirrhosis and irreparable tissue damage. Again, tying into the function of the spleen, they are also involved in removal and recycling of erythrocytes and iron and bilirubin. It produces about 900 mils of bile. So what does that look like? I like to use examples. So your liter bottle of pop, yeah, that's about how much bile you produce per day. So pretty impressive and pretty important. And so what's really cool is that it can regenerate. And so you can actually remove portions and it can grow back. And so if you remove about 70% of it, it can still return back to its original mass, which is why oftentimes when you get transplants, you can transplant a liver to multiple patients because it can regenerate. But you still can't live without it. And so if we look at the microstructure of the liver, it's actually quite beautiful. You can see these hexagonal shapes known as lobules with central veins. And so here's a nice blow up of what that looks like. And so what we're going to do is kind of dissect out what that lobule is and so how the liver actually filters. And this is really nice because it ties into what we were talking about with the capillaries. And while I was on the last exam, we still, again, it's cumulative. And so we're going to talk about the differences between fenestrated capillaries that we see in the kidney when we talk about the urinary system. And so here we're actually seeing sinusoid capillaries, so those really leaky capillaries in the liver. And so if you look at the lobule here, this hexagonal shape, you can see that it's surrounded by veins. These veins are bringing blood from the digestive tract, so blood from surrounding the small intestine, large intestine, stomach, and spleen. All of that blood gets sent to the liver for purification first, and then returns back to normal circulation. And so that's what you're seeing here. So it's called a portal vein. It's part of the hepatic portal system. We'll talk about it again in a little bit just to highlight it. And so any blood from that was surrounding the small intestine, large intestine, etc., goes to the liver. So here we have that blood going in via the portal veins and then passes through towards the central vein. And so the hepatic portal vessels and hepatic arteries bring blood and kind of it gets sent into that central vein via the sinusoids, so the sinusoid capillaries. And so the hepatocytes are those tiny little cells that you're seeing surround here, and so they absorb and secrete materials into the blood to kind of help purify it before it gets recycled or gets sent to the rest of the body. The blood then leaves the sinusoids and enters the central vein, and so then you have lots and lots of lobules, central veins merging together into the hepatic vein, which then enters into the inferior vena cava. And so it's really the flow of blood goes from the outside of the lobule towards the central vein. And so this is in contrast to what's happening with the bile. And so the hepatocytes also produce bile. That bile migrates to the bile ducts and eventually into the common hepatic duct, and then either to the gallbladder for storage or down to the small intestine for usage in digestive processes right away. And so can either go into the common hepatic duct, common bile duct, and duodenum. And so what you can see, again, cystic duct to the gallbladder, is that the kind of movement of purification of blood is from the outside in, and then the bile is the opposite direction. It's from the inside out. It moves to the bile ducts, which are on the outside or the periphery of the lobules. All right, so done with the liver, moving on to the gallbladder. Again, these are very, very importantly linked, right? And so this is one of the few organs where I can say, oh, is it an accessory organ? Oh, can you live without it? This is one of the few ones you can actually say, yeah, you can live without, right? Lots of people live without their gallbladder. If I were to ask you all to raise your hand and how many of you know somebody, almost all of you know somebody or yours personally yourself have had the gallbladder taken out. And really the only major pathology we often see is that you have to be careful of how much fat you consume, right? But some people do have more problematic pathologies. And so it is an important thing to consider. And so what does the gallbladder do? It is a small muscular sac. Hopefully you can all identify it on your cats. And all the models and whatnot. And so it stores and concentrates bile. And so what exactly is bile? Bile is an alkaline solution of bile salts and phospholipids. And so what is the significance of that is that bile emulsifies lipids. And so it's important that I say emulsifies lipids because it doesn't actually enzymatically break down lipids. There's no enzymes involved. And so but what it does is it allows for enzymes to help break it down in the presence of lipases. And so bile does not break down lipids, it just emulsifies them and makes them smaller. And so this is what it actually looks like here. Here you have this large, giant fat globule, and in the presence of bile, salts, and phospholipids, you end up breaking down that fat globule into lots and lots of small globules, allowing for enzymes to more efficiently be able to come in and interact with this surface to start breaking down that fat. And so if you think about it almost like analogous to if you're looking at your mixture of oil and vinegar in your salad, if you shake it up a whole lot, that's emulsifying fat. You're breaking down those fats through mechanical force, right? You're breaking the large fat globules into smaller fat globules. And so during digestion, bile moves from the gallbladder into the common bile duct and empties into the duodenum, which is what you're looking at here. All right, last but not least for the accessory structures, another accessory structure that you cannot live without. We already know and love the pancreas, especially because we've already talked ad nauseum about the endocrine functions, and more specifically the importance of glucagon and insulin in regulating blood sugar, right? And so this is taken care of by the islet of Langerhans, a very specific cell type involved in endocrine function of the pancreas. The pancreas is also very important in the digestive process, and so it does have exocrine functions. And so what it does is it produces an alkaline solution made up of water, enzymes, and electrolytes known as pancreatic juice. And so what this does is allows for pH balance of the small intestine. So if you think about that food moving from the stomach into the small intestine, what is the pH going to be like? It's going to be acidic, right? Because you have a lot of hydrochloric acid being released by your parietal cells. And so when that chyme reaches the duodenum, we need to be able to kind of pH balance it for the enzymes that are working in the duodenum. And so one way to do that is the pancreatic juice is alkaline, also bile is alkaline. And so we have a pH balance helping from the pancreatic juice and the bile. And then it also promotes protein digestion via trypsinogen. And so inactive form of trypsin is trypsinogen. And so we have pepsin being active in the stomach, trypsin being active in the small intestine as the major enzyme kind of breaking down protein. But it also is involved in secreting amylases, nucleases, and lipases. And so a lot of further enzymes to help break down that chyme that's reaching into the small intestine. And so amylases, so that should ring a bell thinking about the salivary amylase. So here we have pancreatic amylases breaking down sugars. We have nucleases, which are going to break down nucleotides. And then lipases, which are the enzymes that are going to start breaking down those fats. And so that's why I have in parentheses require presence of bile, because these lipases to really work efficiently need bile because we need to be able to emulsify the fats into smaller globules. All right, so we're done with the accessory structures, and we're going to move into the small intestine. And so the accessory organs contribute enzymes to the small intestine. It's important to know also that the small intestine has its own set of enzymes that will also complete the digestive process in the duodenum. And so the three regions of the small intestine, duodenum is that very first short region right extending off of the stomach. The jejunum is kind of the middle region. It's roughly 8 feet long. Yes, your intestines are really, really long. And then the ileum is about 12 feet long. So it's the part that's adjacent to the large intestine. And then residence time, again, varies, but around 3 to 5 hours. And so we're going to talk about kind of, as I said before, the general functions and overall features. And so kind of some more specific features. As I said, when I was contrasting the rugae to what's happening in the small intestine, the rugae in the stomach is for expansion. the features that we see in the small intestine, they are for increasing surface area because the major role of the small intestine is finishing digestion and absorption. And so some of the features that increase surface area you can see in these diagrams, we have circular folds, which are deep permanent folds that we see in the small intestine. We have villi, which are these long projections that you can see here. And so the villi are finger-like projections of the mucosa. And then the microvilli are these also known as the brush border. These are microvilli, they're tiny, small projections off of the villi. Again, all of these function to increase the surface area, and so you see lots of microvilli in the duodenum because those microvilli are also really, the brush border has a lot of enzymes that are involved in kind of finishing off the digestive process before food moves across, before those macromolecules move across the epithelium. And so why do we care about increases in surface area? Again, to have absorption be much more efficient. And so what happens in the small intestine, just like we broke everything down in the stomach, again, digestion, we have mechanical digestion, chemical digestion, we have absorption and storage and movement of food, right? And so we have mechanical digestion, we have some movement primarily in the duodenum and maybe a little bit in the jejunum of mixing of digestive juices. So the pancreatic juices, the bile, and the chyme all coming together in that duodenum, in that first region of the small intestine. So we need to mix that chyme and those digestive juices together. We have chemical digestion via digestive enzymes from the liver and the pancreas, and so we have completion of fat digestion through bile and lipases. And so I'm going to fix that slide, but the completion of the completion of fat digestion happens via bile and lipases. We have completion of sugar digestion or starch digestion via pancreatic amylase, and completion of protein digestion via trypsin. Again, when you get into physiology, I'll talk more about the brush border enzymes and the completion of digestion that way, so keep that in mind. I'm simplifying things a little bit. We have absorption happening via the villi and microvilli. Again, we have that ileocecal valve, which I talked about at the beginning, I think, which was joining the ileum and the cecum in the small intestine, large intestine. And so that valve is closed to kind of keep the digestive fluids in the small intestine for as long as they need to be before moving into the large intestine. And then last but not least, we have propulsion via those very important peristaltic waves from the small intestine into the large intestine. And so the digestion is primarily occurring in the duodenum, and then absorption really mostly happening in the ileum, but also happening in the jejunum. All right, second to last check-in, which means we're almost through digestion. So we'll have a check-in here and then a check-in after the large intestine. And so check-in here, so two major functions of liver, what is true about the pancreas, what happens where in the small intestine. So pause and actually try and do this rather than just waiting for me to say it. So the two major functions of the liver, like I said, I listed a whole bunch of functions. And so really it's filtering of nutrient-rich fluid. And so filtering a bunch, there was a huge long list. You can't see me emphasizing that huge long list with my hands, but that huge long list. And so filtering of blood as well as the digestive function of producing bile. And so which of the following is true about the functions of the pancreas? It only secretes hormones. Nope, it does secrete hormones, but it also secretes pancreatic juices. It secretes amylases, lipases, and nucleases. Yes, that is true. It secretes active forms of trypsin. That is actually false. It secretes inactive forms, so it secretes trypsinogen, which then gets converted into trypsin once it's in the duodenum. And that's really important because that way we don't have premature digestion of cells and things like that in the pancreas that are producing the trypsin, right? And so we want trypsin to only be active in the small intestine And so it releases highly acidic pancreatic juices. Nope, it's actually highly alkaline because that's the optimal pH for those enzymes to work. And so then where does digestion primarily occur in the small intestine? It primarily occurs in the duodenum. So where does absorption primarily occur in the small intestine? Mostly the ileum, a little bit in the jejunum. And we all now love the small intestine. All right, last but not least is the large intestine. And so again, features that we've already kind of talked about. The ileum, again, part of the small intestine, but leads to the ileocecal valve, which leads to the cecum. The cecum is that very kind of first pouch of the large intestine. The vermiform appendix sticks across that. We did talk about the appendix in the lymphatics, where we talked about the kind of role of it harboring beneficial bacteria and helping repopulate the gut when you take antibiotics and things like that. And so as we move up, we'll see the ascending colon, transverse moves across, descending down, sigmoid, rectum, and then anal canal, and the fabulous as always external anal sphincter. And so let's talk about kind of what's happening in there and what the epithelial tissue looks like. And so as we've moved through the digestive tract, hopefully you're noticing that we keep talking about the epithelial tissue, and while the major players are still all there, what the epithelium looks like is slightly different depending on the importance or depending on the function of the organ that we're talking about. And so what you'll notice in the large intestine is we don't see any of these intestinal crypts. We don't see any very obvious villi or finger-like projections. You kind of see this almost like flat-like appearance in the epithelium, but you do see lots and lots of kind of white cells. And so you can see them here, all these white cells along here. What this is lots and lots and lots of goblet cells. And so goblet cells, remember, produce mucus, and we've seen goblet cells throughout our entire digestive tract. Again, very important that we have mucus lining our entire digestive tract. But why would we have more goblet cells in our large intestine than pretty much any other structure? Do we need a lot of mucus in our large intestine? Yes, right? So the major function of the large intestine is, of course, water absorption and some vitamin absorption. So what's happening is we have compaction of fecal material. And so as you compact that fecal material, it gets harder. And so you're more likely to have kind of scraping along those epithelial tissue. And so it makes sense that we'd have more goblet cells in the large intestine than we would the small intestine. And so what happens in the large intestine, again, breaking it down to chemical digestion and mechanical digestion, there is no mechanical digestion. In chemical digestion, none of it's intrinsic. It's only via enteric bacteria, and so we talk about the beneficial bacteria. They help us complete some of the digestion, but there's no production of specific enzymes in our large intestine by us. We have absorption of remaining water, electrolytes, and vitamins, and then we have propulsion, so moving food or moving undigestible material into the rectum. Again, residence time is greatest in your large intestine. Maximize that water absorption because it's very important to balance our blood volumes and things like that. And so residence time is about 12 to 24 hours. And so the ultimate goal, again, is removal of any waste material, any material that we cannot absorb, and any material that we need to remove as a byproduct of cellular respiration, or removal of red blood cells or things like that. And so if we look at and directly compare and contrast the small intestine and large intestine epithelium, and so this is just really a slide for your information so you can kind of see the pronounced intestinal villi in the small intestine. You can see these deep intestinal crypts. Here, we do see intestinal crypts, but you can see that the epithelium in the large intestine kind of all one kind of layer, whereas you see these large projections into the lumen in the small intestine. Here you have lots and lots of goblet cells here, and things like that. And so, again, another pause to think about what organ I am from. And so, like I said, we've been talking about the epithelium throughout this lecture. And so where do you think these are from? So I'll give you an answer, or not an answer key, a word bank. The four major structures that we've talked about: the esophagus, the stomach, Small intestine, large intestine. All right, pause and try and think through which tissue is which. All right, so A, what do you notice about, so let's just think about it from an order of where we would see them in the digestive tract. So if my options were esophagus, stomach, small intestine, large intestine, we're going to start with the esophagus. And so what tissue looks kind of resistant to abrasion and not really involved in any sort of absorption or anything like that? Hopefully you picked B. B is surrounded by lots of muscle. You can see that nice thick layer of stratified squamous epithelial tissue. And so B is the esophagus. And then so we're going to look for structures that are then going to be the stomach. And so the stomach, we're going to see kind of the presence of these gastric pits. And so again, a level-ish top of the epithelium, kind of all level, but then these kind of deep invaginations. But we're not looking for an overabundance of goblet cells, right? And so hopefully A is going to grab your attention as looking like gastric pits with some goblet cells, but not an overabundance of goblet cells. Again, with the small intestine, we're looking for the presence of villi. C have very pronounced villi, and then D, we have lots and lots of goblet cells, and so that should scream large intestine for D as the goblet cells. See, isn't histology fun? All right, last but not least is the rectum, and so we have compact, undigestible material moving from the anus to the rectum. It is the last 6 inches of the colon. It is temporary storage of feces. It has an internal and external sphincter. And so it moves from kind of columnar epithelium to stratified squamous with goblet cells. So again, lots of mucus kind of trying to protect those tissues. All right, last check-in, pause, contemplate the world. No, just contemplate the slide and think about it. All right, so where does the greatest digestion occur in the small intestine? Again, this is building on from the previous slides, thinking about most of the digestion is happening in the duodenum in the small intestine, and then the function of the circular folds and villi is to increase surface area. How does chemical digestion differ between the small intestine and large intestine? Well, in the small intestine, we have intrinsic production of the enzymes that are doing the digesting, and so the pancreas is producing various proteases, lipases, and nucleases and amylases, whereas the gallbladder is storing the bile and the liver is producing the bile. And so that's our body's producing it, whereas any chemical digestion that's occurring in the large intestine is the result of bacteria that are living in our gut, and so we are not creating any of those enzymes or anything like that. And so it is the bacteria in our gut that are responsible for chemical digestion in the large intestine. And so which of the following statements are true? A, the small intestine has a greater number of goblet cells than the large intestine. That is false, right? Because we have more goblet cells in the large intestine than the small intestine. B, the chyme reaching the duodenum has partially digested proteins and sugars, and fats have not been digested at all. That is absolutely true, because remember, we have sugar digestion or starch digestion starting in the oral cavity. We have protein digestion starting in the stomach, and so by the time it reaches the duodenum, which is that very first part of the small intestine, we have started the chemical digestive process, but have by no means finished it. C, there are circular folds and villi in the large intestine, and that is not true, right? We just see those intestinal crypts. And then D, compaction of undigested material and water absorption occurs in the small intestine. That is actually primarily the role of the large intestine. And so last but not least is just talking about some of the little features of the digestive tract. And so we've talked about the importance of the portal systems. We talked about the portal system in the endocrine system. So there's a portal system that exists from the hypothalamus to the anterior pituitary, allowing for kind of going through 2 capillary beds to make sure that those hormones that are released in the hypothalamus are going directly to the anterior pituitary and not going to the rest of the body where they might degrade away or fade by the time they actually act on the anterior pituitary. And so the hepatic portal system I was already describing when I was talking about the liver, because remember one of the major functions of the liver is to purify blood. And so any blood that is surrounding or any capillaries that are surrounding the small intestine, large intestine, stomach, and spleen, all of that blood goes to the liver first for purification before it goes to the rest of the body, because we have absorption of material at those places, absorption of macromolecules and whatnot. And so this is known as a portal system because it's those capillaries. That very first capillary bed is, say, the capillary bed surrounding the stomach, or the capillary bed surrounding the small intestine, the large intestine. That's capillary bed number 1. Capillary bed number 2 is those capillaries, those sinusoid capillaries that we're finding in the liver itself. And so that's the second capillary bed, right? And so that's what you're seeing here. Here we have that very first capillary bed. So here's just an example of the capillaries surrounding the large intestine, the stomach, and the spleen. All of these dump into the hepatic portal vein, bringing blood to the liver for purification, and the hepatic veins bringing blood to the inferior vena cava. And so to go over it in general, again, that very first capillary bed are the capillary beds surrounding the stomach, spleen, large intestine, and small intestine. And then the second capillary bed is the liver. Just some other additional tidbits are things like the mesentery. We haven't talked about the mesentery at all, but we've talked about the peritoneum a little bit, the visceral peritoneum and the parietal peritoneum. And so recall that these serosa run together, right? And so they're continuous with one another. And so what's interesting is, when you guys were identifying the kidney to get at the renal artery and vein, you kind of had to dig in the back. And so the kidney is a retroperitoneal organ, and so it's actually surrounded by the peritoneum here. Then the mesentery on this side is actually reabsorbed and lost. And so peritoneum, again, you can see this is a really nice diagram illustrating the continuity between the visceral peritoneum. So here the layer of peritoneum surrounding all the digestive viscera, which is continuous with the parietal peritoneum here. And so continuous serous membrane lining the body wall and the visceral organs. So remember, this is responsible for producing the serous fluid, which makes sure that you have reduced friction. Because remember, as you're moving, your digestive organs and everything else is moving along with it. And so you want those to not be constantly rubbing up against each other and kind of causing abrasions. And so they secrete serous fluid, which reduces friction and provides cushion, which hopefully you remember from, I don't know, maybe the first week of classes. And then talking a little bit more about the mesentery, it's that double layer of peritoneum extending to the digestive organs. And so you saw these in your on your cats, right? So I made sure and pointed out the greater omentum and then the mesentery and things like that. And so these provide routes for blood vessels and lymphatics and nerves. And so it's really fatty tissue. It helps stabilize the digestive viscera in the abdominal cavity and store fat. And so it depends on how fatty your cat. So some individuals had really fatty cats, and so they had this really nice, beautiful, thick omentum. And then there were others that had really thin, kind of friable omentum. And so greater and lesser omentums, and then the transverse mesocolon and mesentery are some of the features. And that is the last summary slide for the digestive system. So I am going to stop talking. And hopefully, if you have any questions, feel free to shoot me an email. What I'm going to do is on Thursday, we'll do a pretty extensive activity just to make sure that we are all on the same page of how the digestive system works and things like that, because I know that it's hard to kind of absorb everything. And so I will post full slides for this. I will also post the abbreviated slides so you can take notes as you're going through the lecture. And then last but not least, I do have these tables that are actually from your book, but I just wanted to put them up here for kind of, they do a really nice job of overviewing what's happening in the different organs and things like that.